The active form of folate in the body is tetrahydrofolate. Forms of tetrahydrofolate are required as coenzyme in numerous essential metabolic reactions. Folate coenzymes act in these reactions as acceptors and donors of one-carbon units, also called methyl groups. Folate coenzymes play an important role in the metabolism of several amino acids, the building blocks of proteins. The synthesis of the amino acid methionine from homocysteine requires a folate coenzyme and, in addition, vitamin B12. Among others, methionine is needed to synthesize a methyl donor called S-adenosyl methionine, or SAM. A methyl donor is any substance that can transfer a methyl group, a one-carbon unit, to another substance. SAM is used as methyl donor at many sites with both DNA and RNA. These methyl groups can silence the activity of genes during development and tissue differentiation, and undermethylation may well result in dedifferentiation of cells. Folate is also involved in the synthesis of DNA and RNA, the molecules that carry genetic information in cells. Synthesis of DNA is especially important in rapidly growing cells, such as red blood cells. Folates are therefore essential for normal cell division and proper growth. Folates are required for the prevention of anemia, a condition in which a person has fewer red blood cells than normal and feels very weak and tired, and also for normal fetal development. Lack of folate can cause neural tube defects in an embryo of a pregnant woman. These can cause devastating and sometimes fatal birth defects. 5 servings of fruits or vegetables per day will provide enough folate for normal embryonic development. However, many women do not eat this amount of fruits and vegetables. To be assured of adequate folate, women are advised to take an additional 400 micrograms of folic acid daily, either as a dietary supplement or in fortified foods. This folic acid supplementation needs to be taken from 1 month before conception until the end of the first trimester of pregnancy. To protect against neural tube defects, the US Food and Drug Administration in 1998 demanded manufacturers to enrich cereals and grains with folic acid. This enrichment increased mean folic intakes by about 190 micrograms per day and drastically reduced the number of babies born with neural tube defects. Folate fortification is also mandatory in several other countries, such as Canada, Australia, Chile, and South Africa. However, some scientists are concerned that provision of extra folate may potentially do harm by raising the risk of certain cancers. This is the reason that many European countries do not mandate fortification of foods with folate. Folate might influence the development of cancer through its role in DNA silencing, DNA replication, and cell division, but the exact way of action of folates is still unclear. Evidence to date indicates that adequate intake of natural folates might reduce the risk of some forms of cancer. However, high doses of supplemental folic acid are correlated with increased colon cancer risk in individuals with a history of colorectal adenomas and increased prostate cancer risk in men. The synthesis of methionine from homocysteine I discussed before is also important to prevent a buildup of homocysteine in the blood. The deficiency of folates in the blood results in increased homocysteine levels. In the 1990s, a number of epidemiological studies identified moderate elevation of homocysteine as an independent risk factor for heart disease. Slightly elevated levels of homocysteine have also been associated with thickening of arterial walls and blood clot formation. Although folic acid and vitamin B12 supplements lower homocysteine levels, recent results from randomized controlled trials indicated that these supplements do not actually decrease the risk of heart disease, but they might provide protection from stroke.